Now let's discuss cell signaling.
Cells need to communicate with each other to coordinate responses to changes in the internal or external environment. Many compounds used as messengers cannot diffuse through the cell membrane or enter through channels or transporters. So how are cells able to respond to these messengers? The message is transferred from the outside to the inside of the cell via membrane proteins. Messengers are compounds produced by cells that alter the behavior or function of cells. These messengers can be classified by how they work or the locations of their target cells. Molecules on the surface of cells can function as messengers. These are used by the immune system and for contact inhibition. Contact inhibition is a mechanism that enables cells to stop proliferating when they contact each other. Cancerous cells lose this ability. Endocrine messengers are hormones that are released into the bloodstream and target distant cells. Paracrine messengers have targets on nearby cells. An autocrine messenger is produced by the cell it acts on. Neurotransmitters are produced by neurons and their targets are postsynaptic cells. Neurohormones are produced by neurons and are released into the bloodstream to reach distant targets. And finally, pheromones are produced by cells of one organism with cellular targets in another organism. Cell signaling occurs in three distinct steps. First is signal reception, where the message is detected by the target cell. Next is signal transduction, or the process of transferring the message from the outside to the inside of the cell. This process alters cell activity and generally involves amplification of the message via the production of multiple products. Last is the effect or outcome of the messenger. Effects may include a change in cell metabolism, enzymes may be turned on or off, it may involve a change in the electrical charge of the membrane, which is important in excitable cells like neurons and muscles, or it could be a change in gene expression. Signals are received by receptors, which are proteins that bind with and respond to messenger molecules. Receptors can be found in the cell membrane, while some receptors are intracellular and can be found in the cytoplasm or nucleus, or can be located on the surfaces of intracellular organelles like the endoplasmic reticulum or mitochondria. We will mostly discuss cell membrane receptors today. Types of receptors include ligand-gated channels, enzyme-linked receptors, or G-protein coupled receptors.
Ligand-gated channels need to bind their ligand in order to open their channels. As you see here, the ligands are bound, which allows the ions to move through the channel. In their typical mode of functioning, ligand-gated channels open to permit these ions to pass through. And the flux of the ions can alter the electrical charge across the membrane. Now let's look at enzyme or enzyme-linked receptors. So we have the ligand, which is the first messenger that binds to its receptor site. And when it binds, this activates a catalytic site on the same molecule. And usually this catalytic site is on the inside of the cell in the cytoplasm. And this enzyme can be used to produce things like second messengers. G-protein coupled receptors are the largest and most diverse group of membrane receptors in animals. They get their name from associating with G proteins, which are a group of molecules that can bind with GTP or GDP. We'll go through how they generally work here, and a video in the next slide will explain it more. So we have the ligand, or the first messenger, binds with the receptor, which activates it. Okay, and then once it's activated, it can interact with a G protein, and which activates the G protein, which can then activate downstream events like activate another enzyme. And we can get production of second messengers.
Many G-protein coupled receptors have a large extracellular ligand binding domain. When an appropriate protein ligand binds to this domain, the receptor undergoes a conformational change that is transmitted to its cytosolic regions, which now activate a trimeric GTP binding protein, or G protein for short. As the name implies, a trimeric G protein is composed of 3 protein subunits called alpha, beta, and gamma. Both the alpha and gamma subunits have covalently attached lipid tails that help anchor the G protein in the plasma membrane. In the absence of a signal, the alpha subunit has a GDP bound, and the G protein is inactive. In some cases, the inactive G protein is associated with the inactive receptor, while in other cases, as shown here, it only binds after the receptor is activated. In either case, an activated receptor induces a conformational change in the alpha subunit, causing the GDP to dissociate. GTP, which is abundant in the cytosol, can now readily bind in place of the GDP. GTP binding causes a further conformational change in the G protein, activating both the alpha subunit and beta-gamma complex. In some cases, as shown here, the activated alpha subunit dissociates from the activated beta-gamma complex, whereas in other cases, the 2 activated components stay together. In either case, both of the activated components can now regulate the activity of target proteins in the plasma membrane, as shown here for a GTP-bound alpha subunit. The activated target proteins then relay the signal to other components in the signaling cascade. Eventually, the alpha subunit hydrolyzes its bound GTP to GDP, which inactivates the subunit. This step is often accelerated by the binding of another protein, called a regulator of G protein signaling, or RGS. The inactivated GDP-bound alpha subunit now reforms an inactive G protein with a beta-gamma complex, turning off other downstream events. As long as the signaling receptor remains stimulated, it can continue to activate G proteins. Upon prolonged stimulation, however, the receptors eventually inactivate, even if their activating ligands remain bound. In this case, a receptor kinase phosphorylates the cytosolic portions of the activated receptor. Once a receptor has been phosphorylated in this way, it binds with high affinity to an arrestin protein, which inactivates the receptor by preventing its interaction with G proteins. Arrestins also act as adapter proteins and recruit the phosphorylated receptors to clathrin-coated pits, from where the receptors are endocytosed, and afterwards they can either be degraded in lysosomes or activate new signaling pathways.
One type of receptor we haven't discussed yet are intracellular receptors. In this example, we have a steroid hormone. And steroid hormones are nonpolar, so they can diffuse directly through the cell membrane using simple diffusion. Once inside the cell, they bind with an intracellular receptor, and the activated ligand receptor complex functions as a transcription factor inside the nucleus to alter gene expression. Once the original message has been detected by the receptor, signal transduction occurs. This relays the message from the outside to the inside of the cell and usually involves amplification of the original signal.
First messengers are the molecules originally detected by the cell that initiate the cellular response. Molecules that relay signals from receptors activated by first messengers are called second messengers. Second messengers activate intracellular signaling pathways that amplify the signal. Signal amplification is an example of a positive feedback loop, which we discussed in the first lecture. Second messenger types include cyclic nucleotides like cyclic AMP and cyclic GMP, inositol triphosphate, diacylglycerol, and calcium ions.
So why do we need second messengers? Well, often the first messenger cannot enter the cell. So the only way to transfer the message to the interior of the cell is through second messengers, as seen in all three of the receptor types shown here. And as mentioned previously, the second messengers are usually involved in signal amplification. This animated slide will give an overview of second messengers depending on different receptor types. So here we have a G protein coupled receptor with the ligand binding, which activates adenylyl cyclase in this case, and then we get production of cyclic AMP, which is the second messenger used in this cascade. Now we have an enzyme-linked receptor. The binding of the ligand activates it. We get production of cyclic GMP as the second messenger, with downstream effects after that. We have nitric oxide. This is a gas that can diffuse directly across cell membranes. And it can activate enzymes that produce second messengers. Here's another G protein coupled receptor. We get activation of it. We get diacylglycerol produced, downstream effects then after that. We can also get production of a different second messenger, an isotyl triphosphate, which causes the release of calcium, which can act as a second messenger inside cells.
To examine signal transduction and amplification, we're going to look at an example using epinephrine binding to a G protein coupled receptor, leading to the release of glucose from glycogen stores. First, epinephrine binds to the receptor, which activates the G protein. The activated receptor can activate several different G proteins. This is the first amplification step. Each activated G protein can activate several enzymes, adenylyl cyclase in this example. Each activated adenylyl cyclase can convert several ATPs to cyclic AMP. The cyclic AMP can then act as a second messenger. The cyclic AMP then activates a protein kinase. Each activated protein kinase then activates a second kinase. This second kinase then activates an enzyme that catalyzes the breakdown of glycogen into glucose. Amplification occurs at several steps in the process. Each activated enzyme can activate many other enzymes, leading to the formation of several product molecules.
This figure summarizes how a single messenger molecule can lead to profound changes in cellular behavior and function via these amplification steps. In this example, we start with one signal molecule and end up with 10 million product molecules. Without these amplification steps, cellular reactions may not occur at all because not enough product could be produced and/or produced quickly enough. As mentioned earlier, second messengers often activate kinases during the amplification process. What are kinases then? They are enzymes that add a phosphate group to another molecule, usually by using a phosphate group from ATP, so energy is used in this process. The binding of the phosphate group changes the conformation of the molecule it is bound to, which often activates the molecule. The largest group of kinases are protein kinases, with more than 500 different ones identified in humans. These kinases orchestrate the activity of almost all cellular processes by creating a functional change in the target by altering enzyme activity, cellular location of the molecule, or its association with other proteins.
Kinases are often named after the molecule they modify. For example, tyrosine kinases attach a phosphate group to the amino acid tyrosine on the target protein. In this cartoon, we see that binding of the first messenger to its receptor activates a second messenger, the activated relay molecule here. The second messenger triggers a phosphorylation cascade that is orchestrated by kinases. This process may seem overly complex, but you get amplification of the signal at each step, and the pathway can be altered depending on the kinases present. So you can get different cellular responses using the same first messenger depending on what kinases are present. Here's another look at phosphorylation cascades from your text that also illustrates how this process is involved in signal amplification. Each kinase that is activated in the first step is capable of activating many targets, and those targets can in turn activate many targets in the next step, and so on. This is an example of a positive feedback loop. If you remember, a positive feedback loop exaggerates a change in a controlled condition. Positive feedback systems also require a mechanism to turn them off. Since signal amplification is a positive feedback loop, it requires turning off once it has been triggered. If it is not turned off, then prolonged stimulation can damage cells. Signal amplification can be turned off by removing the first messenger, which deactivates the receptor. If the process involves the G protein, then the G protein can be inactivated by converting the GTP bound to the active molecule into GDP. The second messenger can be deactivated by enzymatic conversion to inactive forms or by removing the molecule from the cytoplasm. And finally, kinases are inactivated by phosphatases. Phosphatases inactivate kinases and/or their targets by removing phosphate groups from or dephosphorylating molecules. The final step in cell signaling is a change in the function or behavior of the target cell. Cellular responses to a messenger may include a change in cell metabolism. Enzymes may be turned on or off. The electrical charge of the cell membrane may be altered, or it could result in a change in gene expression by turning a gene on or off. In summary, cell signaling involves reception of a signal molecule, signal transduction via second messengers and amplifying cascades, which result in varied cellular responses. The process allows a single messenger molecule to both profoundly change the behavior of the target cell and allows the same messenger to produce different outcomes in different target cells depending on the components in the signaling pathway. If you think about each step as interchangeable puzzle pieces, then you can see that if you have different receptors at the cell surface, then you can get different responses to the same first messenger. In the next step, you could have the same receptor linked to different transduction pathways, like different second messengers or different kinases, and you will get a different outcome or response. Finally, you can have different final targets in the pathway, say one leading to production of an enzyme and another leading to production and release of a hormone or paracrine messenger. This table shows the various ways a common signaling molecule, serotonin, is used in the body. We have one first messenger, serotonin, that can produce different responses in different target tissues. Here we have 6 different receptor subtypes, 5-HT1 through 6. All subtypes, except for subtype 3, are G protein coupled receptors. The 5-HT3 serotonin receptor is a ligand-gated ion channel that allows sodium and potassium to flow in and out of the cell. The other G protein coupled receptors use various types of G proteins in the signaling pathway. This is indicated by the letter G and the next lowercase letter. So the G protein coupled receptor subtypes interact with 3 different G proteins. The different serotonin pathways use two different second messengers, cyclic AMP or IP3. The next two columns show where in the body a particular serotonin receptor is found and the effect the signaling pathway produces. So we have one signaling molecule, serotonin, that can have different effects on different target cells by 1, using different receptors, 2, using a different G protein or second messenger, and 3, if the receptor, G protein, and second messenger are the same, then the second messenger must activate different downstream molecules, such as kinases. As we saw in the last slide, the same signal molecule can have different effects on different target cells because different cells can have different receptors. Or if they have the same receptor, then the next step in the process can utilize different second messengers or kinases, which lead to different actions or outcomes. Researchers have looked at this process and realized that this process is analogous to puzzle pieces that theoretically could be exchanged to create specific desirable outcomes. Rewiring cell signaling pathways could create cells that can be turned on or off to create specific anti-cancer cells, cells that can enhance or inhibit inflammatory responses, or produce specific products like a hormone or a neurotransmitter to help people that do not produce enough of the product. As you can see in this slide, research into designing these customized cells has tremendous therapeutic potential.